Namaste students, Pushpa Vidya. I, JV Dr. Chandrakala, Assistant Professor to Vidyapeeth Women's University, Jaipur. In today's session, we are going to discuss about the biogenesis of mitochondria. The formation of the new mitochondria, like de novo synthesized. According to this hypothesis, the mitochondria arise from de novo from a precursor in the cytoplasm. So, origin from membrane. The hypothesis proposed that the mitochondria the Golgi apparatus of the nuclear envelope and the membrane invaginates and extend into the cytoplasm and tubules. So here it gradually becomes curved and folded and forms a double wall structure of the mitochondria. Okay. So the development of the micro bodies. It is held up the mitochondria as developed by the accumulation of the microbodies in the cytoplasm. So a microbody consists of a single outer membrane and a dense matrix with a few cisternae, which eventually developed into fully formed mitochondria. The prokaryotic origins it is believed that the mitochondria are originated from bacteria. It is supported by many evidences, like the first is the localization of the enzymes of respiratory chain, which in case of a bacteria are localized in plasma membrane, which can compare with the inner membrane of a mitochondria. Okay. The second one, it is in some bacteria, the plasma membrane forms the membranous projections called mesosome, like cisternae of a mitochondria. These mesosomes or the respiratory chain enzymes causes a respiratory chain enzymes. The mitochondrial DNA is circular in its bacteria, and replication process of a mitochondria is similar to that of a bacteria. The ribosome in mitochondria are smaller and similar in size to that of a bacterial ribosome. The chlororampinol inhibits the synthesis of a protein in a mitochondria as well as in a bacteria. Okay. So here the mitochondria depend partially on the mitochondrial matrix and a DNA partially on the nucleus and the cytoplasm of the eukaryotic cells. It exhibits the symbiotic nature of a mitochondria. Replication is held by the mitochondria by self-replicating organelles. The new mitochondria arises by some type of splitting process from the pre-existing mitochondria. So the last hypothesis seems to be probable since the mitochondria have their own DNA and ribosome, they can replicate the new mitochondria. So however, there are a nuclear control over the process of mitochondrial synthesis. Some of their proteins themselves get other form of the cytoplasm of the cells forming under the direction of the nuclear DNA. Function of mitochondria. Here the cell respiration takes place in the mitochondria, and so they are also known as the powerhouse of a cell. They bring about the stepwise oxidation of the food step or a low-grade fuel of the cell transfers the energy and carries ATP and brings about the required activities in a cell. The biosynthesis, active transport, transmission, nerve, muscles contraction, cell growth, and cell division. Mitochondria provide the intermediate for the synthesis and important biomolecules such as chlorophyll, cytochromes, and steroids. Some amino acid also forms mitochondria. Mitochondria actively accumulate in the calcium ions as a calcium phosphate precipitate. They regulates the calcium ion concentration in the cytoplasm by storing and releasing the calcium. The calcium ions regulate numerous biochemical activities in the cell. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur.